Yes, first of all, I want to thank you for inviting me to talk about our research efforts at the Crohn's and Colitis Foundation. And let me start by saying that the Crohn's and Colitis Foundation has been committed for the last 53 years to fund research to understand the causes and the biology of inflammatory bowel diseases, or IBD, with the ultimate goal of finding cures to improve the quality of life of patients affected by these diseases. And while most of the foundation efforts are focused on funding scientists and clinicians in academia and also in the biotech industry, in the last 5 years, we have started to drive internal research efforts as well. In particular, the translational research team that I lead at the foundation is focused on biomarker and precision medicine research. And before I continue describing my research, let me do a parenthesis here to explain what a biomarker is. Biomarker is a biological indicator of a disease state or biological processes that can be measured. For instance, cholesterol in blood is a biomarker that is used to predict the risk of cardiovascular disease. Another biomarker widely used in the clinic is A1C, which is a biomarker of the risk of developing diabetes. These biomarkers are commonly used in the office of doctors and are very helpful for taking treatment decisions. Okay, now with this clarified, I will close the parenthesis and return to the description of my research. My research at the foundation is focused on identifying two main types of biomarkers. First, biomarkers that can predict at diagnosis whether a child diagnosed with Crohn's disease will be a high or low risk of developing complications like fistulas or fibrosis that require surgery. And second, we're also seeking for biomarkers to predict response to treatment with a biologic drug, which are antibodies that are used to neutralize inflammatory factors. The question is therefore, why are we looking for this type of biomarkers? Well, the answer is very simple. Crohn's disease is a very heterogeneous illness with a variable disease course in which some patients have more aggressive disease than others. In addition, about 10 to 20% of patients with Crohn's disease will develop major complications like fibrosis and fistulas that are very difficult to treat once they develop. Generally, these complications require surgery, and recurrence after surgery is very common. Therefore, they dramatically affect the quality of life of patients, and in some cases, they might be lethal also. On the other hand, not only the disease course is variable, but also the response to treatment. About 40 to 50% of patients do not respond to biologics, which are the best treatment available. And about 30% of those responders, they lost response over time. So as you can see, there is a huge unmet need to identify prognostic biomarkers of complicated disease course and predicted biomarkers of treatment response. And therefore, these biomarkers could stratify patients at diagnosis according to disease severity and response to treatment. In this way, doctors could make early treatment decisions to provide the best treatment options, to deliver the right drug at the right time to the right patient, which is the concept of precision medicine. Now, in support of precision medicine, the foundation sponsored for 13 years the RISK study led by Dr. Subra Kugathasan at Emory University and Dr. Ted Lenson at Cincinnati Children's Hospital. The RISK study is the largest inception cohort of pediatric Crohn's disease patients, in which about 1,000 patients with Crohn's disease were enrolled. This is an inception cohort, which means that patients were recruited at diagnosis and before treatment. This makes the cohort ideal for biomarker discovery. The aim of the study was to identify clinical and biological predictors of complications. And in the long story short, the researchers took intestinal biopsies from Crohn's disease patients at diagnosis and found an mRNA biomarker signature of about 80 genes that could predict at diagnosis that a child diagnosed with Crohn's disease could develop intestinal fibrosis within the next 3 years. They also found another mRNA biomarker signature of about 80 genes to predict the likelihood of developing fistulas. This was a great groundbreaking finding that was published in The Lancet in 2017, showing for the first time that analyzing the mRNA of intestinal biopsies, prognosis of Crohn's disease course is possible. However, as you can imagine, developing a clinical test based on multiplex gene expression panel of 80 genes for each complication is quite challenging. So the Foundation Research Department, we roll up the sleeves and embark in a new goal for this study, which is to translate the findings of the risk consortium into an optimized prognostic test to predict complications in pediatric Crohn's disease. And to do that, we started by centralizing all the clinical data and biosamples into the Foundation's biobank called IBD Plexus. Once we centralize all these data and samples, we use artificial intelligence methods to reanalyze the mRNA from patients identified, and we also identify a novel signature of only 13 genes from intestinal biopsies that can predict fibrotic and fistulizing complications at diagnosis. Because this was successful, then we asked the question whether would it be possible to identify prognostic biomarkers in blood. And for this, we used a novel technology developed by the Swedish company Olink that can detect proteins in blood even if they are present at very low levels. We took blood samples from the recent study, collected a diagnosis of Crohn's disease. We ran these samples in the Olink platform to measure the level of about 1,000 proteins. And we applied artificial intelligence analytical methods in collaboration with data analysis company GeneData and identified out of these 1,000 proteins, 14 proteins in blood that can predict at diagnosis the likelihood of a child with Crohn's disease to develop complications within 5 years after diagnosis. We also identified 3 proteins that can predict treatment response, which is defined as induction of corticoid-free remission in patients who do not develop complications. This is very exciting because these biomarkers are found in blood, which eliminates the need for invasive endoscopies and intestinal biopsy collection, as in the case with the mRNA biomarker signature. In contrast, a simple blood sample from a newly diagnosed child could be sufficient to predict disease course and response to biologics if we can develop such protein tests based on our research. The ultimate goal of our research will be to translate these findings into a prognostic test with clinical utility. And to do so, we started by assembling, together with Olink, a prototype protein panel test that combines in a single panel the 14 protein biomarkers for prognosis of disease course and the 3 biomarkers of treatment response that we discovered. We have been very successful in building such panel, and we performed all the analytical validation of this prototype, assessing different quality control measurements. We successfully passed this quality control stage. And what we did next was to run blood samples from the risk study in this prototype panel, and we were able to stratify, once again, patients at high or low risk of complicated disease course using this prototype test. Our proximal next step is to evaluate the temporal stability of this biomarker signature. In other words, can our protein biomarker stratify patients at risk of complications, irrespective of whether the test is done at diagnosis of Crohn's disease or several months after or years after the diagnosis of Crohn's disease? Now, while this is important, our most critical next step is to validate the performance of this prototype test in an independent cohort, different to the risk cohort that we use for the discovery of these prognostic protein biomarkers. We're not planning to build another inception cohort, but rather collaborate with researchers around the world that might have a cohort of pediatric Crohn's disease patients enroll a diagnosis and who can contribute blood samples for our validation efforts. Finally, we're also in the process of submitting a patent to protect these findings and facilitate the work with industry test developers. Now, I'm going to shift gears to talk about parallel efforts that we are conducting. We are working in partnership with the Biomarkers Consortium of the Foundation for NIH, known as FNIH, to identify biomarkers to diagnose fibrosis for the selection and enrollment of adult patients into clinical trials of antifibrotic drugs. And why this is important? As I mentioned at the beginning, about 10 to 20% of pediatric patients with Crohn's disease will develop complications. However, if you look at the entire patient population, including adults, about 50% of Crohn's disease patients will develop fibrosis during their lifetime. Fibrosis is currently diagnosed using cross-sectional imaging based on magnetic resonance imaging, MRI, and CT scans. The problem is that these techniques to detect fibrosis are insufficient as they are not validated or not accurate enough to be reliably used in the clinic. Thus, you can see here another unmet need for biomarkers that can, in a minimally invasive and reliable way, diagnose fibrosis in IBD. So to address this challenge, the Biomarkers Consortium of FNIH is working in collaboration with the Crohn's and Colitis Foundation, and together we have established a working group to identify diagnostic biomarkers of fibrosis. And I'm honored to chair this working group together with Dr. Sean Connolly from Bristol Myers Squibb. The working group consists of several pharmaceutical companies, also diagnostic labs and CROs, including Olink, academic researchers from Cedars-Sinai and Cleveland Clinic, and also the regulatory agency FDA. And the goal of the consortium is obviously to identify and validate fluid biomarkers of fibrosis for adult patient selection and enrollment into clinical trials of antifibrotic drugs. So imagine that if we can identify a blood biomarker that can diagnose fibrosis without the need for imaging, recruitment for these trials will be faster, easier, and cheaper. All right, so how are we going to do this? We will take advantage, once again, of the Foundation's Biobank IBD Plexus. And in this case, we will not use the risk cohort, but the adult cohort called Spark IBD. This is a prospective cohort of 4,300 patients that collects longitudinal patient samples and data, which is very important for biomarker discovery. And what we're going to do, once again, we will be analyzing protein biomarkers in blood. We will be using the proximity extension assay platform from Olink, and we will also use a platform from Nordic Biosciences that detects in blood different forms of cleaved collagen. Collagen is the main component of fibrotic tissue. Therefore, these pieces of broken collagen released in the blood can be used as readouts of fibrotic processes occurring in different organs, like in the intestine. My message to patients with IBD is that I'm very optimistic with the advances in IBD research and the potential for translation of this knowledge into diverse solutions for patients. The Crohn's and Colitis Foundation has been and will continue to drive cutting-edge biomedical research to provide solutions to pressing unmet needs at any stage of the patient journey, for early prognosis of disease course and prediction of response to treatment, to novel differentiated therapies that can change disease course and that can be tailored to the biological and clinical characteristics of patients with IBD in support of precision medicine. The vision of the foundation and that of our CEO, Michael Oso, and our Chief Scientific Officer, Dr. Karen Heller, is to be committed to advance the field of precision medicine which ultimately depends on successful identification of biomarkers and development of tests like the one I mentioned today, and which can stratify the population in different subgroups based on disease course, response to treatment, susceptibility to disease, etc. This will allow clinicians to tailor treatment to these different groups of patients accordingly. I also must say, we're not only driving internal efforts for test development, as in the case of the protein prognostic panel I talked about today, but we also have a venture philanthropy program called IBD Ventures that I oversee, by which we are making investments in diagnostic companies to develop different types of clinical tests. For instance, we are supporting a company to develop a prognostic test to identify adult patients at high risk of aggressive disease course. We're also supporting another company to develop a minimally invasive blood test to evaluate the healing of the intestinal wall in response to treatment, in order to minimize the use of invasive endoscopies. IBD Ventures is also advancing solutions related to novel therapies, devices, and digital health. And I want to finalize by saying that we're also collaborate with all the key stakeholders in product development and biomarker qualification agencies like the FNIH and contract research organizations like Olink and Gene Data to make sure that the cutting-edge research we are conducting and funding can be translated into real solutions in the clinic to address the unmet needs of patients with IBD.